movement. This chapter, included in the unit of human physiology. This is an entire video course on this chapter. It deals with muscle and muscle contraction, various types of joints, and disorders of the muscular and skeletal system. This video is prepared on behalf of whileforyou.com. Types movements. There are three types of movement in a cell and organs. These are amoeboid movement. It is like that of pseudopodia in amoeba, for example, macrophages, leukocytes, and even cytoskeletal microfilaments show amoeboid movement. Ciliary and flagella movement. Ciliary movement in the epithelial lining of the trachea, reproductive tract, etc. Sperms show flagella movement. Muscular movement. Muscles are responsible for most of the movement in a multicellular organism. Breathing, the function of heart, digestion, movement of appendages, locomotion, everything is performed by various muscles in our body. Locomotion is the coordinated movement of skeletal, neural, and muscular systems. Types of muscles. Muscles originate from the mesodermal germinal layer. Muscular tissues have certain unique properties, such as contraction, extension, excitation, elasticity, etc. There are three types of muscles. Cardiac muscles, striated and involuntary, present in the heart. Visceral muscles, smooth and non-striated. They are also involuntary, and support various internal organs, and take part in various functions, such as digestion, reproduction, etc. Skeletal muscles, striated and voluntary, responsible for the locomotion and movement of appendages. Let's learn in more detail about skeletal muscles, their structure, and the mechanism of muscular contraction. Anatomy of muscle fiber and sarcomere. Each organized skeletal muscle is made of a number of muscle bundles, or fascicles, held together by a common collagenous connective tissue layer, called fascia. Each muscle is enclosed by paramoecium, muscle attached to the bone by tendons. See the diagrammatic structure of a muscle.
This diagram shows the organization of a muscle. It exposed the muscle bundle, muscle fibers, sarcolemma and blood supply. We can study the details in the ongoing lecture. A muscle shows the following structure. 1. Fascicles are a muscle bundle. A muscle, such as a biceps, comprises many muscle bundles or fascicles, held together by fascia, a connective tissue layer. Each fascicle contains many muscle fibers. 2. Muscle fibers. Muscle fibers are elongated cells. They are organized into bundles, called fascicles. The characteristics features of muscle fibers are: each skeletal muscle fiber is long, cylindrical and striated. It is syncytium, that is, contains many nuclei. Sarcolemma. It is the plasma membrane of the muscle fiber. Sarcoplasm. It is the cytoplasm of the muscle fiber. Sarcoplasmic reticulum. It is the endoplasmic reticulum of the muscle fiber. This is the storehouse of calcium ions. 3. Myofibrils. Each muscle fiber has multiple myofibrils, running lengthwise and parallel to each other in the sarcoplasm. The alternating dark and light bands, present in myofibrils, give the muscle striated appearance. Each myofibril consists of even simpler structures, called myofilaments. 4. Myofilaments. There are two types of myofilaments present in a myofibril: thin and thick filaments. Thin filaments and thick filaments have two, attached to each other for muscle contraction. The thin filament is made up of a protein called actin. Let us see the structure of thin filament, or actin filament in detail. Thin filament, or actin filaments are made up of 3 different proteins. 1. It contains 2 polymeric filamentous, or F, actin filaments, which are wound together. They are a polymer of globular, or G-actin monomers. 2. Tropomyosin filaments are coiled around the actin filaments lengthwise. 3. Troponin, which is present at certain points on the tropomyosin, covers the active binding sites for myosin. Troponin and tropomyosin regulate the binding of actin and myosin filaments, hence play an important role in muscle contraction. This diagram shows the structure of actin filament. It shows 2F actin made of many globular proteins, shown as light green colored spherical structure. The orange-colored thread-like structure is the tropomesin filament. It runs parallel to F filament. Troponin is represented as reed beads of group of 3 intermittently. Now, let us see the structure of myosin filament or thick filament. Thick filament, or myosin filaments are made up of the protein, myosin. It is a polymeric protein, consisting of monomeric units of a protein, known as meromyosin, MM. It has 3 parts, a tail, a short arm, or neck, and a globular head. The head and cross arm protrude outwards from the filament at regular intervals, forms the heavy meromyosin or HMM. The globular head contains binding sites for ATP and actin. The head acts as an ATPase enzyme. The tail forms light meromyosin or LMM.
This is the diagrammatic representation of myosin filament. The head shows ATP site, actin binding site and short arm, together form the heavy meromyosin. The tail forms the light meromyosin.
Sarcomere, the contractile unit of muscle. A myofibril contains hundreds of sarcomere, which are connected end to end. A sarcomere is the basic unit of muscle contraction. It contains repeating units of actin and myosin filaments, in an orderly fashion. Sarcomeres are joined, at the end by interweaving filaments forming the Z line. It is the limiting membrane of the sarcomere. Actin filaments, or thin filaments are attached to the Z line, at frequent intervals. Thick filaments, or myosin filaments are present, between the thin filaments. They are held together, at their place by a very thin fibrous membrane, known as M-line. The actin and myosin filaments overlap each other, in a specific pattern, and result in the striation pattern of muscles. They form 3 types of bands. 1. I-band, or isotropic band. This is the light band, and comprises actin filaments attached to the 2 adjacent sarcomeres. Here, thick and thin filaments do not overlap. 2. A-band, or anisotropic band. This is the dark band, it contains overlapping actin and myosin filaments. 3. H-zone is the middle portion of the thick filaments, where thin filaments do not overlap the thick filaments. This diagram shows the arrangement of actin and myosin filament. The actin filament, on either side of the myosin filament, which partially overlap the free end of the myosin filament. This arrangement leave a clear zone in the center of thick filament called H-zone.
Mechanism of muscle contraction. Muscle contraction is the result of the sliding of actin and myosin filaments over each other. Sliding filament model was proposed by Andrew and Hugh Huxley. Sliding causes an increase in the overlap of thick and thin filaments and shortening of the sarcomere. This results in the contraction of muscles. Let us see how the mechanism operates in detail. Brain or spinal cord, CNS, transmits a signal through motor neurons for the initiation of muscle contraction. The neural signal causes the release of the neurotransmitter, acetylcholine, at the synaptic cleft of the neuromuscular junction. Acetylcholine binds to receptors present on the muscle fiber and causes depolarization of sarcolemma. The action potential thus generated spreads through the muscle fiber. Calcium ions are released in the sarcoplasm from the sarcoplasmic reticulum. The release of calcium ions is regulated by another protein, dystrophin. The dystrophin-coding gene is the longest gene in the human body. The calcium ions bind to troponin and cause conformational changes. The myosin binding sites present on actin filaments become exposed. Myosin head also contains a binding site for ATP, where ATP binds. ATP hydrolysis takes place by ATPase activity of the myosin head. The energized myosin head, or cocked, binds to the active binding sites on actin, forming a cross bridge. The attachment is followed by the release of phosphate from the myosin head, triggering the power stroke. Myosin filaments bend and pull actin filaments towards the center of the sarcomere, causing shortening of the sarcomere and muscle. ADP is released in the process. The detachment of myosin head is also driven by ATP. In the presence of sufficient calcium ions, the process repeats again and again.
Observe the diagram carefully. See the attachment of ATP, its hydrolysis and pulling of actin filaments. towards each other and the cross arm become in detached condition. This followed again ATP binding and pulling, actin filament. This process will repeat again and again. After the muscle contact maximum, it remain in relaxed condition. Let us see how it happening. Once the neural signal ceases, acetylcholinesterase inactivates acetylcholine in the synaptic cleft. Muscle fibers come to a resting state. Calcium ions are pumped back into the sarcoplasmic reticulum. In the absence of calcium ions, the troponin-tropomyosin complex again covers the myosin binding sites on actin filaments. Said line of sarcomere returns to its original position, and muscles become relaxed.
After many contraction and relaxation, the muscle become fatigued. Let us see how it happening. Muscle contraction is powered by ATP. Muscle fibers get ATP from the storage of creatine phosphate and glycogen. Under normal conditions, the glycogen is converted to glucose, which further releases ATP in cellular respiration. In the case of strenuous exercise, muscles require high amounts of energy. The body could not supplement the oxygen demand, and glucose is broken down anaerobically. This results in the accumulation of lactic acid, the lactic acid causing muscle fatigue. Rigor mortis, and types of muscle based on myoglobin content. Rigor mortis, the temporary stiffening of muscles, after death is caused due to depletion of ATP as the cellular respiration ceases. ATP is required for the detachment of myosin head. In absence of ATP, the cross bridges remain intact in the muscles, which were in the process of contraction. It helps in estimating the time of death. There are two types of muscle fibers, based on the amount of oxygen-binding pigment, myoglobin, present in them. Red fibers, or aerobic muscles, reddish in color, contains more myoglobin and mitochondria and less sarcoplasmic reticulum. White fibers, or anaerobic muscles, pale or white in color, contains less myoglobin and mitochondria but more sarcoplasmic reticulum. The second part of locomotion and movement chapter is skeleton. Let us see the various bones that make the human body. Enjoy the cartoon, to begins the study of skeleton. Let us see the various bones in human body. There are a total of 206 bones present in an adult human being. Bones are hard, due to calcium salts present in the matrix, whereas cartilage contains chondroitin salts. The human skeleton can be divided into the axial skeleton of 80 bones, and appendicular skeleton of 126 bones. Axial skeleton, 80 bones. It includes skull, vertebral column, ribs and sternum. Appendicular skeleton, 126 bones. It includes pectoral and pelvic girdle and limbs. Study the table carefully, and make sure that human skeleton is made up of 206 bones. This diagram shows the skull. Skull includes cranium with 8 bones articulated by suture joint, facial bones with 14 bones and ear ossicles of 6 bones. The skull is composed of 2 sets of bones, cranial and facial, that totals to 22 bones. Cranial bones are 8 in number. They form the hard protective outer covering, cranium for the brain. The facial region is made up of 14 skeletal elements which form the front part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity, and it is also included in the skull. Each middle ear contains 3 tiny bones, malleus, incus, and stapes, collectively called ear ossicles. The skull region articulates with the superior region of the vertebral column with the help of 2 occipital condyles, dicondylic skull. Let us observe various bones of vertebral column. vertebral column is formed by 26 units called vertebrae. It extends from the skull. Each vertebrae has a central hollow portion, neural canal, through which the spinal cord passes. First vertebra is the atlas, and second one is axis. The vertebral column is differentiated into 1. Cervical, 7. 2. Thoracic, 12. 3. Lumbar, 5. 4. Sacral, 1 fused, and 5. Coccygeal, 1 fused. The number of cervical vertebrae are 7, in almost all mammals, including human beings. The vertebral column protects the spinal cord. It supports the head, and serves as the point of attachment for the ribs and musculature of the back. Now, we can study the sternum and thoracic cage formation. It is formed by 12 pairs of ribs, and sternum ventrally, and thoracic vertebrae dorsally. Each rib is connected dorsally to the vertebral column, and ventrally to the sternum. It has 2 articulation surfaces on its dorsal end, and is hence called bicephalic. Ribs are classified into 3 types, as 1. True ribs. First 7 pairs of ribs are called true ribs. Dorsally, they are attached to the thoracic vertebrae, and ventrally connected to the sternum, with the help of hyaline cartilage. 2. False ribs. The 8th, 9th and 10th pairs of ribs do not articulate directly with the sternum, but join the 7th rib with the help of hyaline cartilage. These are called vertebracondyl, false, ribs. 3. Floating ribs. Last 2 pairs, 11th and 12th, of ribs are not connected ventrally, and are therefore called floating ribs. Now, let us see the different bones of forelimbs and hind limbs that form appendicular skeleton, along with pectoral and pelvic girdles. Limb nose. 1. Forelimb bones. The forelimb bones are humerus, radius, and ulna, carpals, wrist bones, 8 in number, metacarpals, palm bones, 5 in number, and phalanges, digits, 14 in number. 2. Hind limb bones. Femur, thigh bone, the longest bone, tibia and fibula, tarsals, ankle bones, 7 in number, metatarsals, 5 in number, and phalanges, digits 14 in number. A cup-shaped bone called patella cover the knee ventrally, kneecap. Phalangeal formula in human is 2 plus 3 plus 3 plus 3 plus 3 equals 14. Pectoral girdle. It is formed of 2 halves. Each half of pectoral girdle consists of a clavicle and a scapula. Scapula is a large triangular flat bone, situated in the dorsal part of the thorax, between the 2nd and the 7th ribs. The dorsal, flat, triangular body of scapula has a slightly elevated ridge called the spine, which projects as a flat, expanded process called the acromion. The clavicle articulates with this. Below the acromion is a depression, called the glenoid cavity, which articulates with the head of the humerus to form the shoulder joint. Each clavicle is a long slender bone, with 2 curvatures. This bone is commonly called the collarbone. Pelvic girdle. Pelvic girdle consists of 2 coxal bones. Each coxal bone is formed by the fusion of 3 bones. 1. Ilium. 2. Ischium. And 3. Pubis. At the point of fusion of the 3 bones is a cavity called acetabulum, to which the thigh bone articulates. The 2 halves of the pelvic girdle meet ventrally to form the pubic symphysis, containing fibrous cartilage. Let us see the different joints in human skeleton. Joints in human skeleton. Joints are points of contact between toe or more than tendons, or cartilage. Force generated by the muscles is used to carry out movement through joints, where the joint acts as a fulcrum. Joints are classified into 3 different types. 1. Fibrous joints. 2. Cartilaginous joints. 3. Synovial joints. The synovial joints are, further calcificento. 1. Ball and socket joint, between humerus and pectoral girdle. 2. Hinge joint, knee joint. 3. Pivot joint, between atlas and axis. 4. Gliding joint, between the carpals. 5. Saddle joint, between carpal and metacarpal of thumb. This diagram shows various suture joints in the cranium, and bull socket joint formed by head of femur and acetabulum of pelvic girdle. This diagram, shushing joint between humerus and radia, ulna, and pivot joint between atlas and staxis. This diagram, shushing gliding joint and wrist, saddle joint between crapple and metacarpal of thumb, and condyloid joint. Now, we can study about disorders of muscular and skeletal system in human. 1. Myasthenia gravis. It is an autoimmune disorder, affecting neuromuscular junction, leading to fatigue, weakening, and paralysis of skeletal muscle. 2. Muscular dystrophy. It is characterized by progressive degeneration of skeletal muscle, mostly due to genetic disorder. Other disorders on muscular and skeletal systems are 1. Tetany. It is characterized by rapid spasms, wild contractions, in muscle, due to low calcium ions in body fluid. 2. Arthritis. It is characterized by inflammation of joints. 3. Osteoporosis. It is an age-related disorder, characterized by decreased bone mass, and increased chances of fractures. Decreased levels of estrogen is a common cause. 4. Gout. It is characterized by inflammation of joints, due to accumulation of uric acid crystals.